vlog, it's Nev, and as you can see, we're doing a little something different.
Hello, I am Sydney, and today we will be presenting for the first time ever, the Barn Biologists.
What does that mean, Sid?
So today we are going to be giving the viewers a chill podcast-style history lesson about GFP as a molecular tag.
Wow, this will definitely stick in my head for a long time. Okay, so I'm going to talk about this in steps because I just find it easier to learn that way. First, GFP, which stands for green fluorescent protein. This bioluminescent protein is naturally found in the jellyfish Equoriae victoriae. I had to note the pronunciation for this one. It brings out our first scientist, Osamu Shimomura, who first purified the famous green fluorescent protein. Take it away, Sydney.
Okay, I like how you say first scientist. There's actually 3 scientists who shared the 2008 Nobel Prize for the discovery and development of GFP. So first was Osamu. He's first from Japan and he came to the US, specifically Princeton, when he was invited to research Equoriae victoriae. And in 1961, September, the purifying process began on a sample of Equoriae victoriae taken from Washington State. And in February 1962, they discovered green fluorescent protein.
Well, this may sound boring, but I feel like it's really cool. Shimomura studied a jellyfish only to find the first thing that can literally glow on its own. When exposed to blue light, of course. Now, I'm going to connect the next link to our chain and introduce the second scientist, Martin Chalfie. He obviously saw value in the isolated green fluorescent protein. I did a little poking around and noticed Chalfie was a biologist at Columbia University, New York, who specifically studied small organisms such as C. elegans.
What is C. elegans?
I'm not even going to try to pronounce the full name, but all you need to know is they are 1 millimeter long and they are transparent roundworms.
Oh, okay. So his thought process was that it would have been easier to see the small cells if they produced GFP themselves.
Yes, exactly. GFP would emit a natural glow under UV without needing to add chemicals or disrupt the cell. One thing I find a little sly is that Chalfie was not the one to locate the GFP gene. A researcher, Douglas Prasher at Woodhull Oceanographic Institution in Massachusetts, actually found and supplied the genome.
A good partnership, I see.
No. A genome trio.
No way!
Yes way! Because I think credit also goes to the graduate student, Ye Yokoshin, who actually succeeded in getting E. coli to produce GFP with him. The GFP DNA sequence was linked to the gene he wanted to study. Scientists could now follow a visual signal of where a gene and eventually protein went throughout living cells. I'm picturing them looking through the microscope and then in 1994 and it's just like a like slow-mo moment, but this cell's literally glowing with GFP.
Mm-hmm. So while GFP was helpful because of its luminescent ability, the light was pretty dim and only came in a bleh kind of green. So another scientist, Roger Chen, decided to study and develop new GFP variants that showed more strength and brightness and could be various colors. This allowed researchers to then distinguish different proteins. So to be simple, he fancified the combined ideas of GFP as a molecular tag, and then he mixed those properties with different colors.
Oh my gosh, I know of that guy. As a proud older sister, I also feel it is my time to say that he was working at UC San Diego, where my little brother goes to school. Anyway, I found a quote he gave the San Diego Union Tribune in 2008 where he said, "Color helps make the work more interesting and endurable. It helps when things aren't going well." I feel that, don't you?
For sure. Before we end this tale, I just also want to mention how cool it was to see the Brainbow projects. You can really see the importance of GFP's discovery through this project.
Yeah, agreed. GFP has enabled scientists to label different tissues, cells, proteins, and organelles, which are so important for studying various biological processes, behaviors, or even expression. But how was GFP used in the Brainbow project?
Basically, after GFP was isolated, modifications were made to it to change the excitation and emission wavelengths, and by doing so, it let scientists tag different things of interest with different colors. So in the case of the Brainbow project, these different GFP variations let scientists label different neurons and then map the brain's neural circuits.
Wow. So this work definitely could not have been possible without the discovery of GFP.
Definitely.
Okay. Well, guys, that wraps up our first podcast video with the Barn Biologists. This episode was on GFP as a molecular tag. Hopefully you guys enjoyed. Like and subscribe down below.